take the chance to talk a bit about biological DNP solid-state NMR spectroscopy, which happened to be my favorite technique. I'm Jiafei Mao from Institute of Chemistry, Chinese Academy of Sciences. And I have to say, actually, my current research interest is on really very different things. So maybe I've not get up everything fully updated, but I hope to give you a good overview. Then you can dive into the literature through here. So, okay. So in this tutorial, we'll touch some key components of biological DNP solid-state NMR spectroscopy. I would like to give away some practical considerations, which could be very important for your projects, and also to set up a starting point of literature diving, which I believe is really, really crucial for, doing DNP studies. So please take this lecture or tutorial on your own risk, and especially under critical thinking, because the field is moving on very fast and many things just cannot get fully updated, as I just mentioned before. So which types of information can NMR provide? So I summarize here. First, we can get a lot of chemistry information. So please remember that NMR is a powerful technique actually in the chemistry field, so we can understand the electronic structures by NMR spectroscopy. But in biofield, of course, we all know it's a powerful method to get to the structure of biomolecular molecules. And not to mention, NMR is super powerful in actually mapping the molecular dynamics, which is still not very easy to be obtained by the competing techniques, I would say. So now let's put for example, memory protein into our the magnetic field. What we see here is actually if you have fully labeled memory protein, in this case, human beta-2 adrenergic receptor, there are just thousands of nuclear spins you can explore. So this very complex situation actually is even more because we can indirectly visualize actually the electrons. As I said, NMR is also a technique to access the chemistry. So basically electronic structures. So we just suddenly have so many spins. And let's take a look at the nuclei. So here are of course, our beloved nuclei you can use in NMR spectroscopy. As we all learn from textbook, from how the higher gyromagnetic ratio is, the better NMR sensitivity you get. So proton is of course the best, and nitrogen will be the difficult one. And now you can put these spins somehow magically on your interested molecule from your proteins and run through this pipeline. And in principle, you will get a lot of information on the chemistry, structure, and the dynamics. But all these are built on the premise that we can visualize each individual spins in your molecule. And that's actually the bless of NMR spectroscopy because we really have the atomic resolution. But in fact, as all kind of analytical technique, you get something, you lose something. And in NMR, what the problem is, although we have this bless on the resolution, we do have a bottleneck, that's the sensitivity. And I think everyone knows this. So to tackle this problem, I want to show you an example. For example, we can use very high magnetic field, in this case, 20 Tesla, 850 megahertz. In that time, we decided to put a memory protein to this spectrometer and get, NCOCX2 diversion. The problem is that even with the gadolinium doping, so now you have very fast acquisition, still we lose a lot of signals because the sample is just occupied by a lot of lipid and water, and the molecular weight is not too small, so we don't have enough interesting molecule in the NMR rotor. That's really the problem because as you can see here, we use 850 megahertz. That's actually one of the highest fields that you can find for solid-state NMR spectroscopy. So there's not too much to improve until, for example, we can find a way to really boost, for example, the gyromagnetic ratio of the point where you started the NMR experiment. So we already see if you start from the proton, then you possibly can't find another nuclei to replace it, but we can start from the electron. So when you add unpaired electron into your sample, now we have a technique called dynamic nuclear polarization. So basically, we use microwave to saturate the electron spin, and then by multiple mechanisms, you can transfer the polarization from the unpaired electron spin to the nuclear spin. So now we see the gyromagnetic ratio of these two spins are very different. So when you pull the polarization from the electron spins to the nuclear spin, you have huge boost of the NMR sensitivity. So this is actually one of the most powerful hyperpolarization technique. And if you want to learn a bit more about the polarization transfer mechanism or possible strategies, I would refer to Monu's tutorial also in this series, in which you will find a lot of details. But today, just to make it at a more practical level. So how to set up a DNP-enhanced solid-state NMR experiment? You need a few components. So you need the radicals, which carry unpaired electrons. So these are the polarizing agents. So you have to distribute the radical in your sample. They relate to sample preparation. And of course, you need a DNP solid-state NMR spectrometer to run the experiment. I would say this is a complicated instrument, but with this setup, you often can get, tens to hundreds fold of sensitivity enhancement. That means thousands to tens of thousands time saving. In some cases, without this kind of a boost of sensitivity, you won't be able to see anything. And on the right side, I've just visualized one example that by adding radical and microwave, that suddenly we have really enough sensitivity so we can start to dive into more complex spectroscopy. So here I give you an example. That's the same sample we tested on 850 megahertz high field room temperature solid state NMR spectrometer and a 400 megahertz relatively low field DNP solid state NMR spectrometer. And you can see that the sensitivity obtained under DNP condition is really superior compared to the normal high field technique. So this is really the way to go if you are first limited by the sensitivity. I would say if you don't see the signal, then there is nowhere to go by the NMR spectroscopy. So first we need to visualize something. The key components, as just mentioned, for solid-state experiments are DNP agents, sample preparation, spectrometer, but also please think about this is called a DNP solid-state NMR spectroscopy. So at the end, we are doing solid-state NMR experiments. So it's really important to think about which NMR experiments you want to set up under DNP condition. I will touch it in an example later. Then you see there are many factors. And obviously, when you start, the question is which to choose. But I would say there are not so many options. You may first think where to go because access is very important. I think the most expensive component is definitely the spectrometer. So first, one need to find a spectrometer on which you can run the DNP NMR experiments. So most of the DNP solid-state NMR instruments are actually made by Bruker. So I just listed here the Bruker's commercial system. As shown in the photo, this is our instrument in Beijing. But usually Bruker provides the field from 400 to above 900 megahertz. They are building really very high field, even higher at the moment. The temperature can go down to 100 Kelvin. So if you use 3.2 millimeter rotor, that's very standard on DNP setup. On Bruker system, you can usually go to 12, or if you take a risk, maybe go to 14 kilohertz at a low temperature. So that's a record actually on the zirconia rotor, I would say. And if you go for sapphire rotor, which is really better for DNP experiments because it's more microwave transparent, and this material has higher heat conductance. But the fact is, sapphire is not so robust as zirconia, so you can't spin very fast. And on the other hand, if you do have the risk of, for example, a rotor crash, which will be very unfortunate, of course. But in that case, you may put a risk on sapphire rotor, I would say, because sapphire rotor crash possibly want to destroy your stator. In most cases, even don't destroy the coil. And if you use 1.3 millimeter rotor, which we also practiced a bit in Beijing, then we can really go to 40 kilohertz at about 100 Kelvin. But the moment what we see is the vespral cap on 1.3 millimeter rotor, at least from Bruker, has a bit of the quality issue, and sometimes it just tend to be broken after several runs. I would mention one point that Bruker's system actually has a lifetime. Actually, the lifetime of the gyrotron, the microwave source, is about 10 years. That's quite long. But if you start a project, you may not want to start from an older system because if the instrument break in the middle, then there are not so many DNP solid-state NMR spectrometers around the world. And your project may be interrupted. So I actually see a number of the cases in different labs. So that surely, I think all the students here, you definitely should consider this factor. Another thing for the method developers, I would say Bruker system has very limited field sweeping capacity. So it's really not very good for exploring, different radicals or, for example, the metal ions used a lot in beyond Cornelius group. So where to find the instruments? The best is, of course, you have one in your lab or in a neighboring lab, but often it's not the case. So I only know, for example, in Europe, also including UK, there are a number of infrastructures you can, apply for. And through these infrastructures, you basically can have machine time covered or even your trip covered. So that would be a very good way to access DNP solid-state NMR technique. I think in United States, there are also national laboratory which can provide the access to many, many users. I would mention actually there are also a number of very exciting home-built systems. I just listed a number of them. They are actually very exciting because can go down to really, really low temperature using helium cooling. So these instruments are not commercially available and are only actually accessible in very few labs. But if you do have a very particular application, especially you need a very low temperature to freeze some weird dynamics, for example, the mesial rotation at 100 Kelvin, then possibly these spectrometers will be the good option. So now if you decide which spectrometer you want to go, you of course know the magnetic field. And that will help you to decide which type of DNP radical you want to choose. So if you stay at 400 megahertz or intermediate field, I would say 600 megahertz like we have in Beijing. And for, of course, biological applications, you need often water-soluble radicals. And you can take from CortecNet, that's the company sales radical, the MUPOL radical or M-tinipol. So I will show next slide, actually, the performance of M-tinipol is better than the MUPOL. It's just slightly more expensive. And another radical I found very popular in the field is asimple pork. A number of labs actually get from collaborators this very nice radical. But if you move to really, really high field, 800 or 900 megahertz, you need possibly the tritol-based radicals. And I would say I do a very, very slim summary here. Actually, there are many, many radicals. Just it will be impossible to cover all the radicals even on one slide. So I just listed as many as groups I can remember who developed the radicals. And if you really want to take a look at this history, you can definitely from here to dive into the literatures. And of course, it's also possible sometimes to find a collaborate to make customized radicals for particular applications. And this, for example, is from my collaborate, Yangping Liu's recent work. It's actually he participated joint comparison, kind of a competition between different radicals. And they did demonstrated that at high field, really this kind of snapple is the winner. So the tritone nitroxide radicals are very likely to be at the moment best option if you are so craving for sensitivity. And if the sample is really so precious, I would say it will be worthy to contact him and get some 10 triple or snapple for your applications. So when you get a radical, the next question is how to put a radical into your sample, which sounds very easy. It's not actually so simple. We have to consider if the radical is stable in your system, which means you cannot use low pH because the nitroxide radicals will definitely die at low pH, no reducing agent in your sample preparation. Another challenge, I think, is the main challenge is how to distribute radical homogeneously. Homogeneously means often at a molecular level throughout your sample. That's really a challenge. You can imagine, actually, we have very limited factors we can play with to reach the homogeneous distribution. Of course, we also have to think about the radical concentration. What I can give you as a tip is usually start from 10 to 20 millimolar for these radicals. They tend to be always optimized in this region. And again, if you have find a similar system in the literature, then just take a look at similar cases and start your optimization if necessary from that records. The experience that we actually accumulated last time in Frankfurt is please store your sample, if possible, at -80. Because if you keep the sample for a long time for the next measurement, and sometimes radical dies at -20 even. So the -80 or even low temperature, if you have a nitrogen tank to store them there, would be very good for maintaining the sample integrity over a long period of your project. So as I just mentioned, that how to distribute the radicals throughout the sample is really a tricky point. And often I find it's the decisive factor of the whole project. So I want to give you the example that I did in Clemens Glauby's group in Frankfurt. That actually we find if you work on proteoliposome, so basically have a pellet, then the best way to deliver the DNP radicals is to use 30% glycerol overnight soaking. So that's actually very unconventional because as you possibly all know from the literature, the standard recipe, so-called DNP juice, contains 60% of glycerol. And I think many labs use 60% glycerol. But in our case, actually, this high concentration of glycerol will destroy proteoliposome. So we optimized a lot and really went down to 30% glycerol. In this case, the sensitivity is excellent. You can see the enhancement is still very good. And the titration, of course, helps a bit, but not that much. So I always feel it's difficult to convince the people to use 30% glycerol when all the literature, or most of the literature, say 60%. So after 5 years, we decided to publish another data, actually also in the supporting information, to compare with Bob Griffin's, the gold standard, that's the bacterial dopsing, on which they optimized the DNP juice, which contains 60% glycerol, and really see that on bacterial dopsing, you need 60% glycerol for the better DNP enhancement. So here, definitely, our result shows the recipe is sample dependent, that you have to consider the feature of your sample to optimize the recipe. So it's actually a bit tricky, but you can see in the field, people know for a long time. So here I show you the Ben Rive's work published in 2011. So that's more than 10 years ago. They already know if you have, for example, the native membrane preparation from bacteria, you don't need a glycerol. So that's Angevante Chimie paper. They get very decent DNP enhancement using amyopol that time. And also later on, Tatiana's group find on their capsid, the best recipe contains 20% glycerol because goes to higher, if you go to higher concentration, the capsid will just be destroyed. So we mentioned a bit the different recipes and talk about, use different glycerol concentration, which enhancement you can get. But in your sample optimization, actually enhancement is not the only factor you would consider, because enhancement is not everything for the sensitivity. So of course we want to better DNP enhancement, then the sensitivity hopefully will be better. If you read at a radical, you also get some help from paramagnetic relaxation enhancement, so you can acquire the data faster, let's say. But there are a number of other negative factors, for example, the paramagnetic quenching introduced by paramagnetic radicals. Some radicals, especially some nitroxide radicals, do show significant depolarization on the magic angle spinning that will actually eat up a part of the sensitivity. Also, the paramagnetic species will shorten the coherence lifetime, and that will be the problem if you want to set up a very complicated recoupling experiment. Another factor, I think, which is kind of straightforward, but it's difficult to recall at the beginning, is that if you add 50% glycerol, you basically reduce the effective amount of your sample by a factor of 2, and that's quite a loss. So just to sometimes think about these kind of things. When you add all this glycerol or some reagents, you occupy a valuable sample volume and reduce the effective sample amount. So here I want to show you again a real world example that's from Clemens' group. They have extensively optimized actually DNP sample preparation in a detergent memory protein radical system. And here you can see by comparing, for example, amyl and asympl POC, very different sample preparation conditions, very different recipes. One really need to combine all the factors to get the best sensitivity per unit time. So that's often really, really tricky. So it's really important to take a look at the literature and to consider if there are still important factors beyond the sensitivity to be optimized in your own case. So now imagine if you have excellent sample preparation, very good spectrometer, and DNP is well set, then we get hopefully very good sensitivity. But as I mentioned at the beginning, you get something, you lose something. And in this case, unfortunately, we start to lose the advantage of NMR, which is the resolution. So the problem is when you freeze the sample to 100 Kelvin, which is often loved by most of the DNP experiments, you start to freeze all the dynamics in your complex biomolecule system. So now your protein does not look like one conformation, but shows the conformational distribution is the static disorder, which will definitely cause line broadening. And this is the case, for example, on this proteorhodopsin memory protein. And when we freeze it, you can see the line get broader and broader. So that's really the problem, that at 100 Kelvin per EM, it actually can resolve very good the structure, but for NMR spectroscopy, we enter the field called blobology. So that's really the problem, and later I will show you a number of examples how we solve this issue. But first, we need to realize that what you need maybe is not a resolution. It showed on the left, of course, the resolution is very important. If you have higher resolution, you can maybe resolve more sites and see more information. But sometimes what we really need is the power of resolving. That's not a resolution, it's a bit different. You can see, as shown on the right side, if you have really terrible resolution, but you only have one or two signals and they are separate, you still have enough power of resolving, although you don't have excellent resolution. That sounds tedious, but this will decide the strategy used for the DNP NMR experiments on complex biomolecules. Because if you don't have excellent resolution, then sometimes it's not possible to do the conventional multiplexing spectroscopy, which means you do, uniform labeling on this protein and to visualize all the sites through one sample. But sometimes you have to just dissect the system to multiple samples, different labeling schemes, and do kind of monoplexing. So that's tedious amount of work. but will be better to, get something. So if you just label everything, in some case you have excellent signal, but you just don't know which is which. At the end of the day, I think this kind of signal is useless. So how to actually do different monoplexing? We just need to play, the spice beans at the intrinsic side. So that's very much depend on your biological questions. And here I just give you a cartoon, some examples how we can play the spice beans on interesting memory proteins. Of course, they all have advantage and disadvantage. So I would suggest you to go through more detailed examples in the literature to see which method you possibly would like to use on your particular system for your particular question. And here I also would like to give you some examples that nowadays we have a number of strategies. You can put the artificial probes, for example, these fluorine probes on your sample, but there are also a number of very nice native probes we can use, like the backbone nitrogen, the cyanide methyl group, and so on. And although we have, the time limit, so I really can't go to all the details, but you can see actually there are really many, many factors coupled together to influence the sensitivity and the resolution in your DNP solid state NMR sample. So they are really, really complicated. And often what we have to do is a lot of compromise. You need to find that balance, that sweet spot for your particular sample, and more importantly, particular NMR experiment for the particular application. And to this point, I guess we are ready for some examples. So I will classify the examples through actual resolution, not the sensitivity. So the case one, what if we have less resolution at low temperature? Imagine if we can freeze the sample and still have very good spectrum. And that's the case you can do multiplexing. And there are a number of this kind of blessed system. For example, Adam has this beautiful, type 2 secretion needle, and they are tightly packed. So the molecular order is fantastic. As a result, when they freeze the sample at a low temperature, actually the order or structural homogeneity is still there. So it's possible to actually do DNP solid state NMR experiments on uniform labeled sample and still run through the sequential assignment. And on the right side, of course, Bob has also a beautiful example on A-beta 42 fibril. In this fibril, as you know, the core parts, they are tightly packed, so they can really survive from the freezing process. Because nevertheless, they don't experience that much dynamics even at the room temperature. So when we freeze down, and they still show the structural homogeneity. So in this case, you basically will have excellent sensitivity and also very good resolution. So you can do experiment really to your favorite condition. But in many cases, maybe your sample will not be so good. So what if the resolution is not good at a low temperature? And then we have to pick out more like a monoplexing approach. So that's one case I actually experienced when I was a postdoc in Clemens group, want to study a peptide bound to a memory protein, a receptor. So imagine there are only 9 residues on this small peptide, but we realized, okay, when we freeze the sample, the line width will be terrible. So we can't just use uniformly labeled peptide to run through the project. So we decided eventually to use 6 labeling schemes to cover 9 residues, which means 6 samples. So that's a lot of work. But the good thing is on each sample, you have all the signal well resolved and you will have very short assignment. I would say actually we don't need to do sequential assignment anymore because they are just so sparse. And in this case, we can run very easy, 2D experiment and assign all the chemical shifts. So if you read this paper, you'll feel it's a bit weird because we actually do nitrogen carbon TDOR, carbon carbon DQSQ experiment. And usually when you do solid state NMR, people like more like the DCP based experiment, like like a carbon carbon PDSD experiment. But here the choice of the experimental NMR method is very special because we realized we have very few sample, which means we can't even do optimization of the matching conditions. I actually tested, we need 4 hours to possibly see the signal from the DCP experiment on the 1D mode. And you know, DCP is not very robust, as shown on the right side. Oskinat and Nielsen group has actually done a lot of work to show the matching condition. And often they are quite narrow. I think if you do solid state and mass spectroscopy, you also know to optimize DCP sometimes it's a bit tricky because you have very, very small window to operate. So in this case, it's not possible to optimize the matching condition. So we have to go back to very simple and very robust method, in this case, the TDOR. Because for TDOR, if you test it, you will see you have a missetting of a bit of RF power or so on, and still the spectrum will work. And that's actually the particular choice of the experiment. And another thing I would like to mention in this work is there are actually two very strong signals in the carbon-carbon DQ-SQ spectrum. So if you just check very carefully, they come from the glass. But imagine in this case, I used a very special glycerol from CRL, that's carbon-depleted glycerol. So you all know there is actually about 1.5% natural abundance C13 in your glycerol or deuterated glycerol. In this case, you can see it's very, very low abundance. But still, because we have so much glycerol, even use the double quantum filter in this case, you would still see the cross peak between the natural abundance and carbon C13 signal in this glycerol background. So if you use normal glycerol, you have a huge signal at this position, and then it will evade some nearby signals and cover or mask your important information. But unfortunately, I have to tell you that actually CIL failed to do one big preparation since several years. So now they actually don't have this product, but they promised if there are enough requests, they possibly will restart the production. So if you don't have the stock, and if you can't get the stock from some collaborators, and you have to be very careful on this aspect when you plan your NMR experiments or the whole DNP project. So the third case is actually about how to get the NMR selectivity by biochemical stringency. As we said, sometimes we have to go to very tedious labeling schemes in order to resolve or visualize selectively some interesting sites. But can we do a bit differently so we can hopefully not go through this pain? And there are a number of interesting approaches, for example, by radical tagged ligand, or nowadays we would call targeted DNP. So on the left side, you would see McDonald's work, where in which they actually tagged a total pole, that's early generation of DNP by radical to a ligand. Then once this ligand binds to the protein, they can selectively visualize this protein. And the nice thing they demonstrated is that in this case, of course, the local concentration of the radical can be very high. So it's not necessary to use a lot of radicals in the system because they are all selectively attached basically to the protein ligand complex. So Gale did actually something similar. In this case, they can illuminate many sites on the protein with relatively good resolution. So that would be actually a very nice approach to try. The only cost, of course, is the synthesis of this radical tagged ligand. That's quite a lot of chemistry to do. And then when I was in Clemens' group, also developed a method starting from the methyl group. They have very weird DNP, solid state and mass spin dynamics. So we can use them together with some sparse labeling scheme on the protein to visualize selective site. So we call this method simple. And as an example, you can see using this CH3 group as a selective torch to illuminate nearby residues, we can have really, really nice selectivity, even in a complex system. Actually, we run through this technique to the cell envelope, can visualize the selective residue in a memory protein where all these residues in all the memory proteins in this case are labeled. But there are even some more selective method. That's fluorine NMR. So as you know that fluorine is bioorthogonal because we don't have native fluorine species in our cell. So here is Tatiana and Angela demonstrated that you can actually do fluorine NMR on the DNP setup and even start to do the 2D versions. So this would be a very, very nice approach, I think, in future to be adapted in many bio-solid-state NMR approaches. So to this point, I think the field is also moving on to so-called in-cell DNP solid-state NMR because that cells are really some of the most complicated and most heterogeneous system. And here I would guide you to two reference protocols. And on the left is the early approach developed by Mark Baldus and co-workers. So it's quite a simple approach that you just use standard GMP juice in that case to incubate the cell, and magically, actually, the DNP enhancement can be obtained. But nowadays, I would suggest you to use more sophisticated approach developed by Kendra. So here, basically, it's a mimic of kind of cell storage or freezing we do in the normal lab. So this programmed freezing really will keep the high viability of the cells and keep everything more native. In this case, they also use the electroporation for the radical delivery, which can, of course, be applied for protein delivery, as Mark already demonstrated in his work. Another nice thing in Kendra's approach is that you only need to use 15% glycerol, so the cells become heavier when compared to very high concentration of glycerol. That's really inducing some stress to the cells. And when you do in-cell NMR, you want to really approach some more physiological relevant condition. So this kind of very, very detailed optimization becomes really necessary. So with this, I think it's already the end of this tutorial. Of course, I wish everyone who want to do biological solid state, biological DNP solid state and mass spectroscopy studies the best luck. Thank you.
Thank you very much for the talk. It was very practical. So where to find and what to choose. So yeah, well now we can open the floor for questions, and I have questions. So yeah, so before starting, because you give an advice, very nice advice for graduate students and people start to work with DNP systems. So as student, like how to know that my system is blessed before starting?
So I would say basically you have to try, right? But according to experience, as summarized already on the slide, if you have really tightly packed molecular complexes, like the needle or amyloid fiber, the core part, they are usually very good even at a low temperature. And another example, for example, on the in retinal proteins. So we also studied retinal protein in Clemens group. The resolution was not fantastic at low temperature, but the resolution on the retinal part is really good. Actually, it was not new because Bob has demonstrated this decades ago when they started to work on bacterial rhodopsin. The reason is because these ligands are embedded very deeply in the core of the protein, so the structural heterogeneity is less compared to the other parts, the outer layer parts. That's also the tip that if you have very well organized binding site, maybe it's very rigid. Yeah. If you are running a project, I think together with MD, that would be the bless.
Okay.
because the MD people can tell you how dynamic this part is, and you can already start to estimate, you can even predict the chemical shift distribution, which will allow you to kind of simulate the low temperature spectral pattern.
Well, okay, so another question actually. So actually, yeah, for carbon depleted glycerol, the C12 glycerol, yeah, we run many NMR spectra, DNP spectra, and we never see actually glycerol signal with the depleted. Like even when we do DAR, for example,
Yeah.
I mean, it's impossible to see, but you saw it in a carbon
Yeah.
spectra, right?
I guess the point is that we had very few labeled peptide in the sample. So if
Okay.
you calculate, you will see it's comparable to the natural abundance carbon-carbon pair.
So all
So
the signal
if
was
you
coming
have a
from.
lot of samples, then that natural abundance background in carbon depleted glycerol part is nothing, right? It's just a noise level. But if you have very few amount of interesting molecule, you will see it.
Okay, gotcha. We have question from Julia. Which new fields are the most exciting for DNP applications?
Well, that depends on your question, actually. And I have to say, I actually removed some slides based on this. So it really depends on what you want to see, right? Because DNP is a technique, as I mentioned here, is part of the NMR. So if you want to understand some chemical feature, electronic structure of the ligand, you can do NMR, right? If you want to see some structure features, context, you can do NMR, or some dynamics, which will be indirect because we have to go through the conformational distribution and then somehow to understand the dynamics. So the tools and the methods are there, but if you only ask about me about the application, the application depends on application itself. That means which maybe protein system or nucleic acid system you are interested in. And the impact, I would say, or significance does not come from the method, but come from the system, come from the function, actually. So you need to have good function. If there's a good function, and you have to derive a structure hypothesis, right? And if this hypothesis can be tested by an NMR approach, that will be the sweet spot. I think the NMR can come in to address this. So it's not on the hand of NMR, but it's on the hand of biology.
Okay, yeah, so you need to find an interesting question that needs DNP.
Yeah,
Okay.
exactly.
We have another question. Can you please explain why deuteration improves DNP enhancement? Is this due to the increased T1, proton T1? How does reduction of proton spin diffusion affect the enhancement?
That's a good question. So in our case, possibly not due to the increased proton T1, but really because we just remove a bit unnecessary proton reservoir. So you have your hyperpolarization goes more to the interested part. That's exactly what Bob has, mentioned long time ago. Because you don't want 100% deuteration, otherwise you spend your hyperpolarization on too many other protons, but you still need to keep some protons, otherwise your spin diffusion will not work, right? So, but the funny thing is, at least on proteorhodopsin embedded in proteoliposome, so this pellet sample, it's really weird that if we do fully protonated system, so that means you add a normal glycerol without deuteration, the enhancement seems very similar. So we don't have really better enhancement when you add deuterated glycerol. But the absolute sensitivity is a bit worse, I would say.
Okay, cool. We have another question. Could you please briefly describe the phenomena of MAS-induced polarizing quenching?
Wow, this actually I would refer to Mona's talk because she mentioned much better than me, I think. The idea that time is, well, of course, so one have to actually take a look at Veda's work and Robert Tico's work. I think they call server effects that time. So what happened is when you do the magic angle spinning, of course, you start to shuffle the energy level of everything. And then there are a number of the matching conditions you can have actually the crossover. Now the problem is you open the channel, of course, to match, for example, the cross effect. That's a three-spin process. I did not mention too much these details in this tutorial. But at a certain point, you also open kind of pathway for the leakage. So it's kind of the two sides of the coin. And that's the place that you start to have the mass-induced depolarization, not polarization quenching, I should say the depolarization. Yeah. And this can be kind of solved by tailoring the radicals. So in simple pocket actually is quite a good one because it shows much less depolarization. Yeah. And there are a lot of theories in there are, I think, some very nice work on the simulation part published in the last years on JPCL. So I will then refer to all these literatures for more details.
Okay, so we have yeah, a question. Thanks for this good presentation. In one of your slides, gadolinium was added to accelerate signal averaging at high field, but leads to missing signals. Is this due to paramagnetic effect? With DNP, stable radicals are added as well. Why, in this case, paramagnetic effect is not the issue?
Okay, so possibly there was a misunderstanding. Yes, gadolinium was the kind of last hope because we want to really accelerate the acquisition, and Bruker made a special probe actually for this E-free fast acquisition. The problem is we still don't have enough sensitivity. So simply when we do NCCX experiment, we can't deliver to more sites because they are just too weak. So that was actually the moment I decided to, give up 850 and move to relay DMP. Because on this kind of a challenging system, we don't have enough sensitivity, even to, you can see this is just a 2D version, right? It's not a real 3D NCCX. It was still very, very difficult in terms of sensitivity limit. And then because gadolinium, of course, did not go into the protein in this case, it's just we did not have enough signal, even with this kind of acceleration. And then the DNP radical, when we added, we did not see on our protein any real quenching. But in another case, in the detergent system, we saw it. So the amine pole somehow a bit concentrated into the detergent micelle. That reduce the coherence lifetime on the peptide bound to the receptor. So I can acquire the TDOR, is shown on the slide, but later we want to do actually a series of TDOR with longer mixing for the distance measurement, and that was not possible because the coherence dies really fast. We can't get the curve. Yeah, I hope that answers the question.
Okay, yeah, great. So I didn't see more questions. And yeah, thank you. Thank you once again,
Great.
Professor, for the fantastic session and for all the practical tutorial, and for you all for your nice questions. Yeah, I think this concludes our session today, and please keep an eye out for our upcoming Global NMR discussion meetings. And yeah, have a nice day or evening, everybody. Yeah. Oh yeah, actually, I have, yeah, sorry, one more question.
Oh,
I see. And
okay. Yeah.
thank you for your valuable talk. As far as I know, the word is doing good in solid state NMR. Can you please explain why word is focusing more on solid state? Oh yeah,
Oh,
NMR.
I love this question. Yeah. So I can't say we are really, I mean, focusing more on solid state NMR. So if you see NMR field, of course, solution NMR is more mature in terms of methodology and also some applications. So when we need to develop a method, solid state NMR has larger space, right? It's a kind of a better playground, I would say. But the solution NMR is actually very, very valuable and super powerful. So I can say from my own experience, because we are in a chemistry institute, and solution NMR can answer a lot of questions, not only the characterization of small molecules, but really we have tons of chemistry questions. I mean, the chemical reaction mechanism or something like that, we have to address by solution NMR, but not solid state NMR. Actually, my lab is also doing a lot of solution NMR these days. So I definitely can't say we are focusing more on solid state NMR. I think on application side, the solution NMR is actually has a big share. It's just sometimes it's too much, and people just say, yeah, it's just solution NMR. But definitely you can find a lot of very interesting niche applications, I think, which can only be addressed by solution NMR spectroscopy.
Yeah, so solution NMR is still the bread and butter of magnetic resonance, and we rely on it. We have actually one more last question from Asif. So the optimization of a simple POC and amipole, right, require different solvent optimization in your experiments. Could you elaborate?
Okay. Actually, that's not my experiment. Johanna did it in Clement's group. Let me try to find back the slide. So I try to explain for Johanna, basically. So the idea at that time is we, of course, optimize the radical concentration, because you usually get from 10 to 20, but want to go a bit low. And the solvent system is a funny story, because I actually at a certain point stopped to use deuterated glycerol in some applications, because normal glycerol is cost nothing, right? And the enhancement, as I said, on the proteoliposome pellet is same actually. So all this information then goes to Johanna, and she had a system which is very precious, so really need some optimization on the model system. That's why here you can see the crazy optimization on the concentration, deuteration, and different recipes. So as if what I can really tell is it's a very special case. Yeah, in this system, we actually Johanna was really praying for sensitivity, a bit of the even better sensitivity. So it's very necessary to use even model memory protein to optimization for her then real world application, which is on a GPCR. But in some cases, when you can't have enough sample, for example, to set up optimization, what we can do is a guess. So all the experience goes to more like 10 to 20 millimolar radical concentration. And the glycerol concentration depend, on your sample character first. You can't destroy the sample in first place by adding glycerol, right? Another thing, what I can tell is on homogeneous system, don't go below 30% glycerol. Because if you go below 30%, then you will have phase separation. And what happen is the ice formation will push out the rotor cap. But if you go above 30, actually the glass will not be perfect, but it's more or less there. So it's already enough to run the DNP experiment. So we actually did a long time ago this test. And the advantage then to use lower concentration of glycerol is that you can pack more sample in the same rotor. So that's a few, I think, boundary condition I can give as a tip.
Okay, one more question from us. How can we know when glycerol destroyed our sample or not?
Oh, that is the point. You have to measure something different. For example, on the cell, you know the viability, right? On a protein, you measure the activity. For example, it's enzyme, you can do the enzymatic assay. On a receptor, we can do the ligand binding assay and some assays. So basically, in the world of biochemistry, it's all about the assays. In the world of material science, it's all about the functional measurements. So depending on which material, you always have a functional readout. And basically you add, your particular DNP juice and see if it interferes with the particular function of your target.